Welcome! Today we'll be discussing recent biochemical research on heat shock protein 104 as a potential treatment for neurodegenerative diseases. As you should know from your biochemistry courses, protein misfolding states can polymerize to form aggregates that underlie several neurodegenerative diseases, as shown here. These debilitating diseases currently affect over 6 million people in the US alone, and incidence will rise as the population ages. Consequently, developing effective treatments is imperative for modern medicine. One recent development is focused on utilizing protein disaggregates chaperones to directly alleviate aberrant protein aggregation as a potential therapy for neurodegenerative diseases. The chaperone we're interested in today is called heat shock protein 104, found in all eukaryotes except for multicellular animals. In 1998, Hsp104 was first discovered in yeast to reactivate heat-damaged protein aggregates. But more recently, Hsp104's activity has been experimentally studied in multicellular animals, yielding promising results that suggest potential utility as a therapeutic agent. Hsp104 differs significantly from the GRO-S chaperonin, which you should be familiar with. While the GRO-S promotes proper protein folding by sequestering proteins to provide a suitable folding environment, Hsp104's disaggregase activity comes from an active translocation of the substrate, but we'll come back to that later. Now, why is this heat shock protein that's found only in unicellular eukaryotes so important? Several studies led by Professor James Shorter in his lab over the past decade have established heat shock protein 104's ability to dissolve various protein amyloids formed in many human degenerative diseases. This has potential uses as treatments for these diseases, so there have been efforts to engineer Hsp104 to increase its effectiveness, as well as to understand its mechanism of disaggregation. In 2014, variants of Hsp104 from yeast were engineered to enhance disaggregation of proteins associated with several neurodegenerative diseases, most notably alpha-synuclein aggregates, which underlie Parkinson's disease. This was done by selecting for various missense mutations in different domains, and one position was determined to have the strongest effect on Hsp104 activity, as shown in the figure on the slide, denoted as A503. The experimenters then mutated A503 to every possible amino acid residue. Mutation of A503 to several residues enhanced the ability of Hsp104 to suppress protein aggregate toxicity. These enhanced variants were able to suppress neurodegeneration in a C. elegans model for Parkinson's disease. This also identified the protein's coiled-coil middle domain as a key region that governs the function of Hsp104, although the exact mechanisms and conformational changes weren't established at the time. In 2017, the structural changes of Hsp104 were finally elucidated with cryo-electron microscopy, revealing the substrate interactions and conformational changes that allow the Hsp104 molecule to dissolve aggregates and amyloids, which I will try to briefly explain. Hsp104 begins in a closed state bound to the folded protein substrate, indicated in yellow, by contact with tyrosine residues. It undergoes a conformational change into an extended state, allowing Hsp104 to move 2 amino acids along the protein. In one cycle, hexameric Hsp104 will thus translocate 12 residues. This conformational change is coupled to ATP hydrolysis by nucleotide binding domains. It was shown that the middle domain exerts allosteric control via an interaction with the nucleotide binding domain, which explains the earlier findings in 2014. The combination of ATP hydrolysis and protein substrate binding triggers a final conformational change in Hsp104 from an open to a closed conformation, releasing the substrate protein and coupling it to encourage proper folding. As we've seen today, Hsp104 has shown exciting efficacy in animal models of neurodegenerative disease. The future direction of this research is to further optimize these systems and potentially develop drugs based on these protein disaggregases. In fact, another disaggregase protein, IgG3P, is now in clinical trials for treating Alzheimer's disease. This demonstrates a novel, real-life application of biochemical topics that you learn about in class.